Thank you very much. Oh, thank you very much for that kind introduction. Thank you all for the opportunity to be here today. And I know we're getting into the evening there in Germany, so I'll try and avoid putting anyone to sleep. So whenever I get the chance to talk to an audience talking about phenotyping or phenomics, I find it is useful to talk about the many different disciplines that have to fit together, because we all tend to mean something a little bit different when we talk about high-throughput phenotyping. We just heard a wonderful talk that I think focused a lot on the engineering challenges of how do we actually design and collect data from experiments to enable biological studies, as well as some of the biological analysis. There's this second challenge of, once you have sensor data, how do you process that into quantitative measurements of plant traits, as we heard, and then how do we use these quantitative measurements to identify genes controlling traits, genotype by environment interactions, that sort of thing. So I bring this up to explain that I'm not going to spend a lot of time on the engineering challenges. A lot of the data I will be showing you today comes from the high throughput phenotyping facility we have at the University of Nebraska-Lincoln, which is a style that will be familiar to many folks in the audience. Plants on conveyor belts that are moved around the greenhouse, weighed, watered, and then taken through a series of imaging chambers. Our system is somewhat uncommon, is that it's designed for much larger plants. So we're able to take grain sorghum and many maize inbreds through their entire life cycle on the system, because we can grow plants up to about 2.3 meters tall. But otherwise, what I'm showing you is work that really could be done at a number of facilities around the globe. This facility generates both RGB conventional images of the plants, which we can take from multiple angles, as well as thermal IR and fluorescence, which I won't be speaking about today, and then a hyperspectral camera that allows us to capture a great deal of difference in the reflectance patterns of individual portions of the plant. Now, before I get into the more recent analyses, I want to highlight some work from Jikai Liang. He was my first PhD student, actually, when I was hired at the University of Nebraska-Lincoln. And when we first gained access to the data coming off of this high-throughput phenotyping system, Jikai was handed the challenge of, okay, how do we process these images and start to estimate properties of the plant? In this case, what you see is a very simple algorithm he developed for estimating biomass from looking at the size of the plant in photos. And it works reasonably well. I mean, it's an R value of 0.9, which means about 80% of the total variation in biomass is being predicted from this simple pixel area counting approach. But Jikai didn't stop with just, okay, this works reasonably well. If we need to, we can run some more replicates. He asked the question, where is this error, the residual, coming from? Is it random or is it systematic? And what he found in this early study was more than half of the total error in biomass estimation from images was explained by differences between genotypes. Some genotypes were being systematically overestimated and some were being systematically underestimated in terms of their biomass. And when we think about it, this makes sense. You could envision different reasons this might be the case, different ratios of leaf to stem, where 1 pixel of stem probably represents substantially more biomass than leaf, differences in leaf architecture between different genotypes, things like that. But I bring this up because I think it's an important thing to keep in mind in evaluating any algorithm for extracting numerical measurements of plant traits from different types of sensor data, but also because more recently we had the opportunity to collaborate with a very talented scientist, Yan Su, and a group at Iowa State University. And there demonstrate that not only is error in measurement of plant traits using high phenotyping methods under substantial genetic control, but we can actually perform genome-wide association studies and map specific genes that are controlling not the trait we're seeking to measure, but our error in the ability to measure that trait. In this case, what you're seeing is work on phenotyping of maize tassels. And what Jan was able to do was actually identify genes that are controlling error in measurement of either the tassel length or the central spike length. And then we can go back and look at the effect of these genes and see some of the phenotypes that are generating this error. For example, having the more erect tassel generates a much more difficult to measure tassel than the more open one. So we get systematic error from that. So we need probably better and more intelligent ways of processing image data that measure plants more like a human being walking through a field or a greenhouse with a ruler and a notebook. And there are different approaches to achieve this. One of these is to actually use sensors that perceive the world in very different ways than we do. What you see here are hyperspectral images collected from an individual sorghum plant. Actually, each of these is one of the individual wavelengths that would make up the hyperspectral data cube. This is work from Chanyong Miao, a recently graduated PhD student in my lab, now employed at Bayer. He and an undergraduate, Alejandro Pages, used a crowdsourcing platform to generate close to 10,000 annotations of individual pixels as belonging to either not plant or within plant to 3 classes, leaf, stem, or panicle. And from this could train computer algorithms to conduct semantic segmentation and actually understand not just where are plant pixels in the image, but what organs do these pixels correspond to. And what you see here are the differences in reflectance profile between these different classes, and then the semantic segmentation of the individual image. And you can see this segmentation isn't perfect, but even the errors kind of make sense. So there are these stem pixels that are being identified here, which are the thick lignified midrib of the plant leaf, which would make sense. It's going to look more like a stem than the main leaf blade. And so using approaches like this, which are using a camera that sees the plant in a very different way than the human eye does, and training the computer to segment the plant in a very different way from the way a human would be able to segment the plant, but using less data, 10,000 individual annotated pixels, or I think actually 8,000 in this case, versus the tens of millions of images for a lot of the more advanced AI. We can then start to measure plants more in the way that a human would. So in this case, we can measure to specific developmental landmarks rather than to the top pixel of the plant. And it also begins to become possible to extract multiple traits from different images. So this is usually the image I show to demonstrate the potential to process sensor data and extract many different traits from a single set of sensor data, at this point a hyperspectral data cube. But I do want to highlight the last speaker mentioned being able to extract 56 different traits from an image of a root mass of a plant. So this really is a general property of the types of image and different sensor data collected from high-throughput phenotyping. And both of these are very different from the way we would normally collect data in a field or greenhouse where human labor is expensive. So we tend to only measure one or a small number of time points, and we tend to only measure the traits we're immediately interested in for that experiment rather than all the traits that it's possible to measure from the sensor data. So as I mentioned, with this hyperspectral measurement, we can collect time series measurements. We can look at how the height of a plant changes over time. We can fit non-parametric curves to that data. Working with a statistician, Yuhang Xu, we were able to conduct an approach called functional principal component analysis, where we can take these different growth curves fit to the change of really any trait over time. In this case, you're looking at height, and look at what are the patterns and how these curves vary with respect to each other. In this case, we identified really only 2 functional PCs that explained a large majority of the variation. The first PC is essentially taller versus shorter. The second PC has to do with how rapidly height increases during the emergence of the sorghum panicle. The nice thing about this functional principal component analysis is we get a score for each individual plant. And from this, we can actually conduct genome-wide association studies and identify genes controlling these different patterns of change over time. In this case, we identify the 3 known genes that are involved in, have major effects on plant height and sorghum, as well as some additional hits that were not previously identified. And the fact that we get 3 that we know gives us more confidence in the ones that we did not. Also, we can dig into the literature and see which genes were identified and associated with which PCs make sense based on the different effects that these genes have on different parts of sorghum height. And then I said the second big difference we see in terms of the types of trait data that come out of high-throughput phenotyping is that they tend to be multidimensional. We can measure many different distinct numerical traits from the same sensor data. When I show people this image, often the question I get is, okay, could we measure leaf angle from this? Leaf angle is, of course, a very important trait involved in being able to pack more plants into the same amount of land efficiently, which is where a lot of the increases in maize yield have come from over the years, is the not more grain per plant, but more plants per hectare. Unfortunately, this is actually very challenging to measure in 2 dimensions. This is a picture of the same plant at the same day, simply taken from 2 different angles. And what you can see is that the actual leaf angle, the leaf angle you would measure from these 2 images is quite different, just based on the positioning of the leaves in 3D space. So in order to measure this, we instead need to actually have an understanding of the plant in 3 dimensions. We were fortunate to collaborate with a very talented computer graphics expert, Professor Benz from Purdue University. His expertise is actually in generating the plants you might see in video games and how to make these plants as realistic as possible. He and his PhD student were able to work with the image data we had generated, conduct a simple segmentation of each photo taken from a different angle, and from this get many segmented views of the same plant from different angles, then use an approach called voxel carving to generate crude 3D reconstructions of plants in space. Now, voxel carving is an approach that has been around for a long time. The real challenge in this is that in our imaging system, and I believe a lot of the systems that are currently being used, photos are not taken simultaneously. Instead, the plant is rotated. Leaves are very thin planar structures that can move slightly. And so a straightforward voxel carving approach tends to actually remove all of the leaves. And so the real innovation in this manuscript was the ability to compensate for that so that we were still able to reconstruct leaves, even with the leaves moving slightly as the images were taken around the plant. From this 3D reconstruction, we can start to phenotype, first of all, just sort of bulk properties of the plant. What you see here is a derived phenotype looking at the size of the shadow cast, so essentially the total amount of light that is being intercepted by individual sorghum genotypes relative to the total volume that they occupy in space. And you can see this is a plant that is very efficient at casting a large shadow for relatively small volume. And this would be an example of a plant that is relatively inefficient. It has a large 3D volume that is not intercepting as much light. And from these traits, we're able to conduct GWAS. We can identify genes controlling these 3-dimensional properties of the plant. And again, this was described a couple of years ago in Matthew and Chen Yong's paper. I should also mention, I mean, this is the real challenge I found sometimes to collaborate with computer scientists is understanding differences in incentive structure. So for Matthew, it was enough if he had a paper where he could demonstrate his technique would work and find 5 or 10 plants that didn't have a lot of problems and were easy to reconstruct. So I was very fortunate in this collaboration to be able to explain and understand and find a good connection on the challenges of going from, okay, we can run it on 5 plants, to how do we actually get scalable code that is going to be easy to run over and over again and robust to errors and challenges and actually process, in this case, close to 400 plants and generate these 3D reconstructions and then calculate the derived traits from these. But once we have these reconstructions, we can again start to do semantic segmentation, organ instance segmentation. What you see here are identifications of individual sorghum leaves from this 3D reconstruction, again, using just, in this case, 5 RGB or 6 RGB photos. So no LiDAR point clouds, no depth information, none of the other cool engineering solutions, but instead a computational solution to get this reconstruction. From these 3D reconstructions, Matthew was able to develop a skeletonization approach where we can actually measure the angle of each leaf with respect to the stem. And from this, we get 1 leaf angle value for each leaf. I'm showing you now his work from Michael Tross, a current PhD student in my lab, using more than 1,000 unique sorghum reconstructions. So this same set of 366 plants, but looking across multiple time points in development. Michael was able to conduct GWAS using essentially the aggregation of all this data. When he does this, we can compare. So in sorghum, what you see here, the yellow triangles are known QTLs controlling leaf angle in sorghum. These were generated in real populations. They aren't mapped to individual genes. What you see here are the positions of sorghum orthologs of maize genes that are known to control leaf angle. And when we conduct a GWAS, we find, among other things, the one gene that is actually known to control leaf angle in sorghum, as well as a number of other signals that correspond well to published QTL and may provide more mapping resolution, although we're really waiting for the resequencing data where we can narrow down where these signals are actually coming from. But this isn't the full potential of the data. We actually have the measurements of individual leaves. So Michael also was able to conduct separate genome-wide association studies for genes controlling the angle of the first, second, and third leaves in sorghum. What you see is some of the signals are consistently identified across multiple leaves. Some are identified only on one leaf, and many of the ones identified only on one leaf do still correspond to known sorghum QTL or orthologs of known genes that control leaf angle in related species. Now, I'm not here to tell you that each leaf is actually controlled by a separate genetic architecture. What we have to keep in mind is genome-wide association study is an approach that is developed to minimize false positives. So if we're able to collect multiple measurements from multiple leaves, we're likely sampling different subsets of the total set of true genes controlling leaf angle in sorghum, and something that is possible because we're able to extract measurements of many different traits from the same sensor data. Now, I want to be careful about not going too far over time. I know we need to catch up a little bit, so this next part is going to be a little bit compressed, which is talking about this analysis challenge of once we really do have measurements for many different traits, how can we best make use of that? Is it really as simple as conducting GWAS for each trait independently and comparing them? I don't think that is the case. So this data is actually from community association panels. It's not from high-throughput phenotyping, but it has the same properties as we are now starting to get from high-throughput phenotyping data, where it's possible to extract 3 or 10 or 56 traits from a single large phenotyping experiment. One approach is this approach pioneered by Chikai in my lab called the genome-phenome-wide association study, where instead of trying to link individual genes to individual traits, we try and identify genes that are linked to phenotypic variation per se across many different traits, and then get a candidate list of individual traits that they are associated with. We can then validate this looking at genes with known loss of function phenotypes. In this case, this is the anthraear1 gene in maize. And what you see here are the phenotypes that are supported in the literature, either from the well-characterized loss of function mutant or previous association studies. And these are the sets of traits that this gene was associated with in the genome-phenome-wide association study, using a set of about 260 distinct trait measurements. You can see many of these are well validated. There are some additional traits that we could not find support for in the literature, although it is unclear whether these represent false positives or traits nobody would have thought to look for in a mutation that primarily affects the development of the maize inflorescence. This is another example from sorghum. This is work from Ravi Maral, a current postdoc in my lab. He was able to assemble more than 230 traits scored across the same population in multiple environments. From this, we can show simple things like increasing marker density actually allows us to identify a larger number of true positive signals. So in many cases where populations have not yet been resequenced, there's probably still useful information being left on the table in terms of genes associated with traits that we can mine by being able to go back to those populations as denser marker sets are generated. But more interestingly, Ravi was able to use an approach called MASH-R to actually look at the effect of individual loci on a wide range of traits. So we can start to get a sense of the potential pleiotropy of loci that we're identifying, which can be useful both in terms of prioritizing individual genes for validation with reverse genetics for integration into a breeding program, things like that. So building these really dense data sets by hand, by people growing plants and walking through the field, is a long process. It can take more than a decade. With high-throughput phenotyping, though, I think we have the potential to do this substantially faster. So one approach to this is what I like to term grow once, phenotype forever. This is work from Michael and also Marcin Wysbowski, who is a postdoc in my lab, and our collaborator Yufang, which is we can go out and collect sensor data from large replicated diversity panels in the field for only a couple of experiments. But over time, what Marcin showed is we can train new models to predict new traits, in this case from hyperspectral reflectance data. And we can go back and accurately extract new traits from this same hyperspectral reflectance data in the past, conduct GWAS, and actually identify genes controlling these new traits. So rather than having to conduct a new field study each time we want to study a new trait, we only have to conduct a small study sufficient to train algorithms to measure new traits from our existing type of data. And then we can reuse these studies over and over again, and we can share them with other people interested in other biological questions. And we can even do this retroactively in some cases. This is a collaboration with Professor Sankaran from Washington State University. At this point, there are commercial companies with satellites orbiting the globe that will happily sell you satellite data of your field trials with resolution as low as 30 centimeters per pixel. If you do this in advance, it's wonderful. You can design your whole study. What we showed in this collaboration was that it was possible to modestly predict yield from the satellite data, even in 2-row yield plots. So this is maybe a 0.75 meter by 4 or 5 meter long plot. It's a quite small area, and the resolution in the data we get from satellites is quite useful. But the motivating thing about this is we didn't decide to do this study until a year after the trial was collected. These same satellites are orbiting the globe all the time. There's very little marginal cost to taking the images. So the companies that provide these services tend to run the cameras constantly, even if they don't currently have a customer. So it's often possible to go back and buy data from your own previous field trials or your collaborators' previous field trials, and again, get new life out of existing studies. So with that, I just want to highlight many of the data sets that my lab uses. We try and release publicly because we want to encourage new people to come up with new things they can extract from these. So this includes the 2D images, 3D voxel structures, and skeletons for the sorghum diversity panel, which are released on Zenodo, as well as hyperspectral reflectance and ground truth data for more than 1,200 maze plots. And then we're always posting new data sets online. With that, I'd like to thank the outstanding folks that I work with, all of you for your attention, and I'd be happy to take any questions if time permits. Thank you.
Thank you very much, James, for this wonderful presentation. And that raises, I suppose, a lot of questions or comments. Mark has one.
So James, thank you very much. I enjoyed your talk a lot. So I would have a question towards the voxel reconstruction. So I'm amazed that you can use video game technology to reconstruct the plant, and you can obviously do GWAS on that. So how much precision is inside these traits? So what is the repeatability or heritability of these traits?
That is a very good question. And do I have that on the slide? I don't think I do. So for aggregate properties like this, amount of shadow cast for number of voxels, my recollection, which I will check and maybe get back to you, is it is relatively high. So perhaps a repeatability of around 0.5 if we look at replicates of the same genotype in the same environment, but separate plants. For traits like leaf angle, it is lower, on the order probably of 0.2 to 0.3 for an individual measurement. And so that is why we found it was really useful to actually aggregate across multiple time points. So simply conducting multiple reconstructions of the same plant on different days, in this case over the course of about 2 weeks, we get substantially higher repeatability across multiple plants than we do looking at only a single time point. Because yeah, the reconstructions are somewhat noisy.
Thank you very much. Very interesting.
Yeah.
Thank you. There is a question in the chat by Marco from Keygene. Thanks for the talk and your work on leaf angle. Did you also take a look at leaf angle variability over time, perhaps under stress?
That's a very good question. At this time, no, we have not. So this study was conducted with an existing sorghum diversity grow-out that we conducted a couple of years ago. I would be fascinated to look at stress. The challenge really is the cost of generating data. So at our facility, it costs about $2,000 a day to run plants on the system. So conducting a new experiment typically requires federal funding. It's not something we can do for an individual project.
All right, thank you. Kerstin first.
Yeah, thank you for this highly interesting talk. I have actually two questions. The first is relating out of personal interest to what, how exactly did this citizen science approach work? Maybe can you elaborate a little bit more on the conditions, what images, how they were named? So how did they really work with this data? And the second is how long did it take to establish all this pipeline to really be able to have this 3D reconstruction for one species, and how fast do you think you can adapt it to various different species? So how much work would you need to calculate per species?
Very good question. So the first about the citizen science platform. So we use an online tool called Zooniverse, which is really good. There are lots of tools out there for doing annotation. So again, I found sometimes it is a challenge with collaborators. It takes a little bit of work to figure out how to upload data into Zooniverse versus just standing up your own tool, but it does a lot of the other stuff very easily for you. Tracking images, repeating. Not for this, but for leaf counting, we actually would show the same images multiple times to people to confirm that how accurate was the data that we were seeing. So I was very happy with the platform, and it makes life really easy. So what somebody would see is essentially what you see on the screen here. They would click each of these and then click 10 places in the image and then click done and move on to the next plant. The one challenge, so Zooniverse, I think, is if you go to the website, a lot of the examples are things like having a camera next to a watering hole somewhere in Africa, and it will ask, well, what are there any animals in the photo? And if so, is there a zebra or a giraffe or something like that? And those tend, I think, to be much more appealing to people doing citizen science than annotating plant images. So in many cases, what we end up doing is when we have a rain out day from the field, I and the undergraduates and graduate students in the lab will actually sign in and be some of the citizens doing citizen science, just because we still have work to do in terms of cracking how to make people excited about plant biology research. So that was your first question. The second one was about adapting the 3D reconstruction. So in terms of closely related species, so I mean, this was developed in sorghum. I think it would work just as well in maize. We're working on doing some validation of that right now. As we think about more plants with more dissimilar architectures, I'm not sure this approach will scale in the same way. So if you think about a soybean plant, which is the other big crop here in Nebraska, or really rice or wheat, there are many more internal voids that may not be visible from any angle that you take photos around the plant. And that's really what allows this approach to work well in maize and sorghum, is if you have side views and top views, you can basically see all of the space that the plant is not filling. Whereas a plant that's more like a soybean, there's lots of empty space that won't be visible. And so for that, I think it will be require fundamentally different analysis approaches, unfortunately.
All right, thank you very much. I've seen several hands being raised. Meanwhile, are there more questions here?
Hello, James. This is Hendrik Porter speaking. I was very much enthusiastic about your idea about growing once and phenotype forever. But as an old-fashioned ecophysiologist, I used to remember that there were also many talks about physiology. And physiology is, of course, a pretty challenging thing if you do non-destructive measurements.
Yes.
So is there still scope for physiology, or where do you hit the wall because you can't go any further with your non-destructive measurements? I mean, I like that you try to combine different traits with each other, but in the end, they are most structural traits. Yeah, where does your approach stop, or where is it so much hindered that we need to develop other techniques? I was wondering.
I think that is a very good question. In some cases, it can be hard to say because many of the things that we're really interested in, simply generating the ground truth data to train a model is quite challenging. So at this point, I don't know if the challenge is that this approach will not scale, or if the challenge is that we can't generate the ground truth data in the first place. So I think of a lot of photosynthetic parameters. Marcin actually tested a number of these, but we just simply weren't able to generate large ground truth data sets. So I think for a lot of things involving photosynthesis, probably transpiration, these are going to be very difficult to measure from straightforward hyperspectral reflectance, as are traits that have strong diurnal cycles. So the collection of this data is a process of hours or days. So we've looked at some things like measurements of non-photochemical quenching. We can actually, I think, get reasonably accurate measurements of that particular plant at that particular point in time. But the challenge is simply that if you collect over 1,600 plots, a lot of the variation you're seeing has very little to do with genetics and have to do with the change in the time of day, whether there's a cloud moving over the sun. So for those things, I think we still need real physiologists. We're not going to be able to replace you folks with machine learning algorithms, which is probably a good thing.
Thank you.
Right. Yeah, Mark. And then we should.
So James, it's me again. Just one question which came to my mind, seeing your satellite images. So are there any legal restrictions, any copyright things you break if you just download images, for example, from your competitor or something?
Yeah. As far as we've been able to identify, no. Now, it may be that, it may be that it would be some surprise. From a purely practical point of view, it is very unlikely you'll have your competitor's field layout, so you almost certainly can find their field, but knowing which plots are planted where really limits the utility of the data unless it's with somebody who is actively collaborating with you and would like this to happen. Right now, in the US anyway, there is a legal limitation. This 30 centimeter resolution is not based on technical ability, but based on what it is legal to sell commercially. If that changes, my anticipation is we could see resolution drop by 1/2 to 2/3, which would make this data much more useful, but that is a legal barrier.
Okay, thank you very much.
Right. Oh, Mitch, you have raised your hands. Thank you.
James, I was wondering about G by E. If we're going to phenotype forever, do we have to have a dozen experiments that we phenotype forever to capture G by E?
Yes, I think is the answer. And I mean, that's your lab is probably one of the handful in the US that really has the capability to do real G by E studies where you're actually growing many different sites to answer questions. So this is one of the things I think actually would be very powerful about this approach is it would lower the barriers to entry to people who are interested in the G by E for different traits if we're able to generate consistent trials across a substantial number of environments.
All right, thanks. There's one more question in the chat by Felix Aikens or Arkens. I got two maybe quite different questions. Have you already worked on finding eQTLs, and do you think working with eQTLs could help finding heritability in complex traits?
It's a very good question. So the answer to the first part is yes, we're doing some work on that now, which is still, I would say, at a medium preliminary stage. What is fascinating, as Mitch was alluding to with the G by E, is we can either look at expression data from the same diversity panels generated in very different environments, or from tissue samples actually collected from the plants in the field where we also collected the ground truth data. There was a preprint that came out just in the last couple of weeks, though, that is doing some modeling and suggests that many significant and real eQTLs may be in genes that aren't directly linked to variation in sort of whole plant phenotypes. So I'm very interested in EQTLs, but one of the things I've seen actually in my own lab is we do an EQTL analysis and we don't find an EQTL for the gene that we think we should based on GWAS for a trait, and we get discouraged. And I think probably we should move away from that. I think there are going to be a lot of genes that don't have detectable EQTL but do have effects on whole plant traits. And there will be a lot of genes that have EQTL, and that EQTL is real. It's real variation in regulation, but it's being detected because it's at a high minor allele frequency and a large effect, because it's not directly impacting fitness in the same way. That was a long answer, but it's a topic I think about a lot.
All right, thank you very much. I would have also points to discuss and or potentially come back to you, because the idea of potentially leaf specific QTL or QTL affecting a particular leaf is also an idea we came up with some of our experiments.
Oh,
I would love to go further on that. But maybe that's we have a different occasion for that. And let Mitch present his talk now. And I would like to thank you once more for your excellent presentation and the very interesting discussion. Thanks a lot.